Okay, thank you so much for inviting me. I'm very happy to be here. And if the last decade, as Dr. Level pointed out, was the decade of psoriasis, I'll convince you today that I think the next decade or this decade is the decade of eczema. Not just eczema, other inflammatory skin diseases such as alopecia areata and vitiligo that will follow. So my disclosures, I did receive a research support consulting or lecture fees from basically I think any company that is interested now in atopic dermatitis, but I don't have any financial gain from any product. And just a very brief overview of my talk, I'll start very briefly with characteristics of atopic dermatitis and therapeutic challenges and opportunities. The hypothesis that atopic dermatitis, similar to psoriasis, is immune-driven and reversible, and the possible contribution of the specific cytokines to the disease phenotype, and then the most exciting emerging treatments for atopic dermatitis, and how do we extend this translational revolution that we saw in psoriasis, we now see in atopic dermatitis to other inflammatory skin diseases such as alopecia areata. So atopic dermatitis, and many of us are dermatologists here, and we know this is actually emerging now as the most common inflammatory skin disease. It used to be considered psoriasis, but now we know it's atopic dermatitis. We are talking in the United States about 4 to 7% of the adults. This is really a huge number, and 15 to 25% of the children worldwide, 15% of the children in the United States, and 25% of the children in Asia. And I can tell you that similar to psoriasis, a third of these are moderate to severe. So very large population in need and very large unmet need for safe and effective treatments for both adults and children. Because what do we have available right now for atopic dermatitis for patients that are severe? Cyclosporin, like you heard from Dr. Lebon, methotrexate that doesn't work so well, Cellcept, and phototherapy. Exactly like what we had for psoriasis about 15 years ago, and in psoriasis we went a long way. So when I started my journey, I started at Rockefeller at a lab that, as you know, actually focuses on psoriasis. So it was natural for me to start by comparing these two inflammatory skin diseases. And as pathologists, you can appreciate, and these are frozen sections, that these diseases look fairly similar in frozen sections. Both have a acanthotic epidermis, and when you stain for the proliferation markers K16 and Ki67, you have wide distribution in the epidermis that is really similarly distributed in these diseases, and the epidermal thickness is very similar in these two diseases. Now, next we went on and looked at inflammatory cells, both T cells and dendritic cells that I cannot show you here. But you see that very large T cell infiltrates characterize both diseases, and it's both CD3 and CD8. Here I show you just CD3, both in the epidermis and the dermis. And the same for dendritic cells. So I asked the question at that time, can the psoriasis model of bedside to bench pathogenic dissection and the very successful translational testing of therapeutics be also applied to atopic dermatitis? And I will try to convince you today that the answer is yes. However, before we do that in atopic dermatitis and in general in inflammatory skin diseases, we have several requirements for a translational medicine approach. We need a well-defined cellular or molecular disease phenotype and very good understanding of immune circuits, and that was not available at the time I started. We need good biomarkers of disease activity, and also, even more importantly, of successful therapeutic response. And I will argue that this is much more important in atopic dermatitis than in psoriasis, because as we know as clinicians, atopic dermatitis is much more difficult to quantify clinically. The lesions blend with the surrounding, they are not as demarcated as psoriasis. And importantly, psoriasis has placebo responses of only 3%. In atopic dermatitis, we are talking about 20 to 23% due to the lack of demarcation. And of course, we need access to drugs that will selectively target the immune system. Now, to complicate things even more in atopic dermatitis, we have two proposed mechanistic hypotheses for the disease, the outside in and inside out. And let me walk you through them. Outside in suggests that atopic dermatitis is a disease of fixed or genetic epidermal barrier defect, and this may trigger abnormal keratinocyte hyperplasia and the secondary immune activation. And this was very largely supported by the finding of the filaggrin gene mutation in 2006. And I want to point out that this mutation is only in 10 to 50% of the patients. In the United States, only 12% of the patients, and only 30% of the patients in Europe. So definitely this mutation is not the whole story, and let's not forget that this mutation also characterizes disease like ichthyosis vulgaris. Now, I will focus my talk on the second hypothesis, the inside out, and this one suggests that the abnormal epidermal phenotype in the lesional skin of patients with atopic dermatitis is driven by increased expression of polar cytokines produced by distinct T cell subsets. Now, just bear with me with this slide. We need to ask the question, what are the cytokine effects on the epidermis in atopic dermatitis? So, these induce epidermal hyperplasia, and this is IL-22 that is derived from Th22 cells. The increased expression of S100s, and we are talking about IL-22 that synergizes with IL-17 for this extent, and they inhibit terminal differentiation of keratinocytes, and these are the Th2 cytokines as shown elegantly by Dr. Leong, IL-4, IL-13, IL-31, and also we showed that IL-22 can do that with potential for feedback hyperplasia. Recently, it was shown that the Th2 cytokines induce key features of atopic dermatitis, spongiosis, and lipid synthesis. And Dr. Leong, in a very important paper in the New England Journal of Medicine, showed that the Th2 cytokines inhibit synthesis of antimicrobial peptides. Recently, it was also shown that the Th2 cytokines augment TSLP secretion by keratinocytes. So we can argue that maybe these hypotheses are not exactly mutually exclusive, and maybe they can be combined because the Th2 and Th22 cytokines could link the barrier and immune defects in atopic dermatitis. So Dr. Lebel showed you this slide of mine, and if you need to take home one message, I think this is the message. It doesn't start from the barrier, it actually starts from the action of cytokines that get increased from non-lesional skin to acute disease, and then furthermore to chronic disease. So what happens here? We have the Th2 cytokines, IL-4 and IL-13. They inhibit antimicrobials. They disrupt the barrier, decreasing filaggrin, loricrin, and other terminal differentiation markers and lipids. And we have the IL-22 that starts the onset of the hyperplasia, and together with IL-17 starts the production of S100s, and also participates in the disruption of the barrier. And like Dr. Lebel said, we have the IL-31 or the itch cytokine that starts the itch, scratch, and lichenification cycle, and all this carries through to chronic disease. And of course, this has a very big relevance to therapeutics that are developed now. So how do we test this immune hypothesis that I put forward to you? A prediction of the immune model is that immune suppression will reverse the epidermal pathology, and this hypothesis can be rejected if immune suppression is achieved but the epidermal phenotype is persisting. And I'll show you briefly two tests of this hypothesis. Our cyclosporin A study and our study with Dupilumab that is an immune antagonist. So we all know that cyclosporin A is effective in atopic dermatitis, is a broad immune antagonist, and we did a study just to see what happens in tissues when you suppress with cyclosporin, because we knew that cyclosporin works in atopic dermatitis. So for this study, we had 21 patients, but 2 dropped out. We winded up with 19, 17 were responders by SCORAD 50. SCORAD 50 is a measure of response, exactly like EASI 50. And if you look at the improvement from baseline, at week 2, there was already 51% improvement in the SCORAD, and at week 12, 72% improvement in the disease. Now, we did in this study that was really difficult to achieve with no funding and 6 biopsies, 2 lesional, non-lesional at baseline, 2 lesional, non-lesional at week 2, and lesional, non-lesional at week 12. So we did 6 biopsies, and here you can see reduction in all the immune axis, since cyclosporin A is not specific, as Dr. Lebel pointed out. So the Th1 axis is suppressed, as seen here with interferon gamma and CXCL10. The Th2 axis is suppressed, as seen by reduction in IL-13 and its chemokines. And the Th22 is suppressed heavily, and also the Th17. And already by week 2, you see major suppression, and basically we know that cyclosporin A usually acts fast on patients. Now, we also obtained, similar to what was seen in psoriasis, a major histological resolution in the hyperplasia following cyclosporin. So this is the baseline lesional skin. As you see, it's very acanthotic. And already by week 2, the lesional skin looks much thinner, And at week 12, it looks really like non-lesional skin. And K16 marks all lesional samples at baseline, and only 2 of the lesional samples maintain K16 positivity at week 12, and already by week 2, many lose their K16 positivity. And if you look at mRNA expression, major reduction in K16, that is 6.7-fold changes at week 12, and also in the epidermal thickness. And this is a more granular view of a transcriptome analysis done by gene arrays. And these are the non-lesional samples at baseline, week 2, week 12, and the lesional samples, week 0, 2, and 12. And red marks activation, blue marks downregulation. You can see that even in the non-lesional samples at baseline, there is some activation, particularly in patients that have higher SCORAD, higher disease activity. And at week 12, this disappears. And basically at week 12, the lesional and non-lesional look very similar. There is overall a 90% reduction in the disease phenotype as marked by lesional versus non-lesional differences. And if you look at the dysregulation in atopic dermatitis between lesional and non-lesional at baseline, they are very wide apart and they come much closer together towards week 12. So the cyclosporine results, you'll agree with me, support the immune model because we did achieve disease reversal both clinically, histologically, and the epidermal phenotype. And this was correlated with suppression of the inflammation. However, although it's highly suggestive of the immune model, cyclosporin A still has direct anti-proliferative effects on keratinocytes, as we know. So this cannot be a final proof of the immune hypothesis. What do we need next? Next, we need clinical trials with specific immune antagonists to determine the specific contributions of the different cytokine pathways, Th2, Th22, and the IL-23, Th17 pathways to the disease phenotype. And I will show you such an inhibitor, and this is Dupilumab that you heard a little bit from Dr. Lebel, but this was the first study. So Dupilumab is a fully human monoclonal antibody that targets IL-4 receptor alpha, and because it targets the receptor, it indeed blocks very effectively both IL-4 and IL-13 cytokines. And this actually started, it was a phase 1b, it started as a safety study, but it was so effective that it was extended beyond that. So this phase 1b study had 67 patients, and Mount Sinai was one of the sites, and 18 of them participated in the biopsy study. And this was a 4-week study with weekly injections of Dupilumab in several doses, 75 milligrams, 150, 300 milligram, and placebo. And these are the dose response of the patients in this 4-week study. And it was quite impressive because the higher doses, the 300 and 150, already are significant at week 2, as can be seen here. And this was a monotherapy study. Unlike the study that Dr. Lebel showed you that was together with corticosteroids, this was monotherapy. Steroids were not allowed. And at 4 weeks, this starts to look like the psoriasis results of maybe 7 years ago. 71.4% of the patients in the higher dose achieved EASI 50 and 54.5% in the intermediate dose. And I want to briefly present you the results of our mechanistic study that involved 18 of these patients. So I told you that K16 is a very good marker of epidermal proliferation. In this study, unfortunately, we did not have immunohistochemistry, so this was our only way to measure improvement in the epidermal hyperplasia. But there is a very nice dose response here in K16. In fact, the reduction in K16 at the end of treatment, which was 4 weeks, is 10.2, which is higher than with cyclosporine of 12 weeks, and this is quite impressive. And this is a map of many markers that we performed by real-time PCR, many measures, and we looked at multiple axes. And what was interesting to us is that, yes, you do get wide decreases in Th2 markers such as CCL26, CCL18, CCL13, and CCL17, but actually the larger reductions are actually in Th17 and IL-23 induced genes such as IL-23A, that is P19, S100A12, elafin, that is an IL-17 induced gene, and K16 itself that is there. And this again is a granular view of a transcriptome. We did not have many patients, but you can clearly see with the 300 that red turns into blue in the 300 milligram. With Dupilumab 150, more mixed results. Placebo really didn't move. Red remains red and blue remains blue. And this is the average improvement in the transcriptome. Dupilumab 300, 52% improvement in the disease phenotype. 150 milligram, about 25% improvement. And with placebo, we have actually an exacerbation in the tissue of 21.5%. And why is this exacerbation important? I just told you that placebo responses in atopic dermatitis are relatively high. And these are the easy responses, the clinical responses of the study, of the biopsy study and of the parent study, and they were very similar. So indeed, placebo responses with 23% improvement, but I just told you that in tissues there was an exacerbation. So if you look at the differences, the clinical differences between arms, 150 versus placebo, it's only 20%. In the transcriptome, we get a 46% difference. 300 versus placebo, 45% difference in EASI, but 74% difference in the transcriptome. And this is why I think it's very important in atopic dermatitis to couple studies with mechanism. So to conclude on that, Dupilumab is the first targeted treatment in atopic dermatitis that does demonstrate dose-dependent clinical and molecular disease suppression. It now finished phase 3, and we are now analyzing actually the mechanistic results of phase 2. This data establish IL-4 and IL-13 as major pathogenic cytokines in atopic dermatitis. And as I told you, larger, longer studies are underway to evaluate the long-term suppression with Dupilumab. And I want to present to you such study that is a monotherapy study of a 12-week given in Europe by Thomas Bieber. And here you can see really results that simulate psoriasis several years ago. The EASI-50 results of Dupilumab 300 milligram are 85.5% versus placebo, that is high but not as high, 36.2, and EASI-75, 61.8 versus 14 in placebo. And this is my own patient that sent me her own pictures, and she is on the extension study of this. And this is how she looked before. These are not as significant actually as what I saw in clinic. And she's a photographer, and this is how she looked after. After only 16 weeks, she looks better now. Still, I have to tell you, there are several things that are not explained by the Dupilumab mechanism of action. The marked reductions in the expression of S100, in the IL-23, IL-17 genes, and the hyperplasia-related genes, and this still needs some work. One possibility is through the inhibition of IL-4-induced differentiation of dendritic cells. So now is the place to ask what features of atopic dermatitis may be explained by other cytokine pathways, IL-22 and IL-23, IL-17. So we and others published that IL-22 promotes hyperplasia and also impairs terminal differentiation, as you see here. And what about targeting this axis in atopic dermatitis? Since IL-22 is involved in the epidermal hyperplasia and the barrier defects in atopic dermatitis, we hypothesized that anti-IL-22 treatment might be effective in chronic AD patients. And we are conducting an NIH NIAMS-funded trial with an anti-IL-22. It's a Pfizer drug in patients with moderate to severe atopic dermatitis. And 2 weeks ago, we actually enrolled our last patient. So we enrolled all 60 patients, and dosing will be completed by the end of the year. We are very excited. And we are blinded, but this is one of my patients in the study. And this is how her lichenified lesions, and you'll agree with me, extremely lichenified, looked before the study. Dr. Level told you that we see the results not as fast as with Dupilumab, but some results are striking. And this is how she looked when she came to my clinic 3 months after the trial. And she still doesn't need any other treatment. So we'll see. Maybe we have the world's best placebo, but maybe we have a good drug. So what is the potential significance of IL-23, IL-17 to atopic dermatitis lesions? IL-23, we know, induces Th17, as you heard from Dr. Lebel, but it also induces Th22. So this axis of IL-23 and IL-17 is also important to induction of S100s and to the epidermal hyperplasia in atopic dermatitis. And we also know from some publications that IL-17 induces S100s, but also impairs differentiation in keratinocytes. So what is the potential significance of this axis to atopic dermatitis lesions? A very important paper from Kenji Kabashima in Japan in mice showed that IL-17 may be required to induce Th2 differentiation. And as I showed you, Dupilumab treatment induced large modulations unexpectedly of IL-23, IL-17 pathway genes in the skin. So what about targeting this axis? I think this axis is clearly expressed in atopic dermatitis and might have therapeutic potential. We just completed a placebo-controlled study with Ustekinumab in 32 moderate to severe atopic dermatitis patients, and we are now analyzing the results, and my lab is also analyzing the Japan Ustekinumab study. And the role of IL-17 itself remains to be determined through anti-IL-17 targeting strategies, and I'm happy to say that we are starting actually at Sinai a single center with Secukinumab in October, and we are very excited, the 44-patient study. Now, this patient was not in one of my studies, but she was treated in my clinic, and she was refractory to cyclosporin and other treatments. I treated her with Ustekinumab, a little bit reducing the intervals, and a great success, and she still is maintained with Ustekinumab. So to summarize for atopic dermatitis, it's not that simple. The phenotype of atopic dermatitis cannot be explained like psoriasis by a single cytokine pathway. We know that psoriasis is IL-23 and IL-17 driven. Atopic dermatitis is more complex, and clinical trials with the different cytokine pathways that will be coupled with mechanism will shed light on the relative contribution of each axis to the disease phenotype. And what are other investigational agents? PDE4 inhibitors, oral and topicals. Oral, we have Apremilast, showed promise in a small investigator-initiated study, and it now is completing phase 2 for atopic dermatitis. There are also a few topical studies. Anacor completed phase 3. Targeting Th2, we have two anti-IL-13 that are in phase 2. Anti-IL-31 that you saw Dr. Lebel's slides, the EAT cytokine completes phase 2. Anti-CRTH2 in phase 2. Anti-TSLP receptor completed phase 1b. We didn't hear results, so probably not as effective. Anti-IL-5 mepolizumab was not effective in clinical trials for atopic dermatitis, but these trials were short, only 2 weeks, and they were about 10 years ago. Targeting IgE, omalizumab, I didn't have time to go into that, but targeting IgE is not helpful for atopic dermatitis. And JAK inhibitors, there are few topical JAK inhibitors that are under development or in early trials, and there will be also several systemic JAK that will go into atopic dermatitis. Now, just very briefly, I want to show you that we can extend the revolution that we have in psoriasis and now in atopic dermatitis to other inflammatory skin diseases, particularly alopecia areata. So alopecia areata is a major problem, lifetime prevalence 2%, usually affects the scalp, but we have patients with total body alopecia. 10% of the patients progress to alopecia totalis or alopecia universalis, and very, very few treatments for alopecia areata. Really devastating disease. And alopecia areata is highly associated with atopy. In fact, a big study from Abrar Qureshi showed in more than 2,000 patients with alopecia put atopy in general and eczema in particular as the highest comorbidity associated with alopecia areata. And there was an important GWAS study that showed that IL-13 was the most associated gene with alopecia areata. And as you know, there are now some studies showing or focusing on Th1 interferon gamma axis, but some other important studies from the past, actually some from Dr. Duvik's group that is here, show that also there is an increased Th2 axis both in the blood and the skin of alopecia areata. And I want to point out that the greatest risk factor for alopecia areata was shown to be atopic dermatitis. So this prompted us to perform a study to profile inflammatory cytokines and pathways in alopecia areata. The study is just published, and this is the first extensive genomic profiling that is published of alopecia areata in a large cohort, 27 patients. So we had 27 patients, 3 patients with extensive alopecia areata, 2 with universalis, 1 with totalis. And we also compared to 5 normal scalp samples, and also compared, and that's very important to compare with atopic dermatitis and psoriasis skin samples. So I just want briefly to show you that indeed there are increases in interferon gamma, and I'll walk you through this. This is normal skin. The red is lesional skin, so here we have lesional psoriasis, non-lesional psoriasis, lesional atopic dermatitis, non-lesional atopic dermatitis, and lesional alopecia, non-lesional alopecia, and normal scalp. So interferon gamma is definitely increased in alopecia areata, but it's similarly increased in atopic dermatitis and psoriasis. And here we have CXCL9 and CXCL10 that are two chemokines associated with interferon gamma. And they are again similarly increased in all these skin diseases. In fact, in atopic dermatitis, we have large increase of CXCL9. But look at IL-23. So both P19 and P40 subsets are heavily increased in alopecia areata, similarly to the other two diseases, and the same with P40, significant increases in lesional alopecia areata compared to non-lesional, and the Th2 cytokines, IL-13, significantly increased in alopecia areata, actually very similarly to atopic dermatitis, while psoriasis, as we know, does not have a Th2 pathway. Now, we also found that alopecia areata, a hallmark of lesional alopecia areata skin, is very wide decreases in hair keratins, and these depend on the scalp involvement. The more involvement you have of the scalp, the alopecia universalis and totalis, the more decreases in the keratins that you have. So to conclude, alopecia areata was found, and we also have gene array studies, I didn't have any time to go into it, is a highly inflammatory disease with increased Th2, Th1, and IL-23 cytokine circuits. It's not as simple, I'm not sure we can just say that it's Th1 disease. Alopecia areata might present a similar model to atopic dermatitis, where the cytokines suppress formation of hair keratins, like in atopic dermatitis, that they suppress terminal differentiation. And clinical trials are very important for targeted therapeutics against the different axes that will test the possible pathogenic contribution of these pathways. And I'm very happy to tell you that we are starting at Mount Sinai several large trials for testing of both broad and specific targeted therapeutics for alopecia areata, and all of them we will do a single center at Mount Sinai. Thank you so much.